we are rolling. Okay. Like I said, guys, what we are talking about today only is the process of DNA replication. And in order to do that, I wanted to first review the parts that make up the DNA molecule. DNA consists of, DNA is made up of a bunch of nucleotides. Nucleotides are like the building blocks of a DNA molecule, and nucleotides are made up of 3 parts, as you guys should already know. You have a sugar here, and if it's DNA, what's that sugar called? Deoxyribose. Okay. You have this over here, which is referred to as what? Phosphate. And then you have the heart of DNA, which is this thing, which is called what? A base. Just call it a base. It's a nitrogenous base, just because it's a base with nitrogen in it, is the only way they get that. Now in DNA, this base can be one of 4 things. As you guys already know, it could be an A, T, C, or G, which stands for adenine, thymine, cytosine, or guanine. In other words, there could be an adenine molecule here, there could be a thymine, there could be a cytosine, there could be a guanine. Okay? That's the basic unit, the fundamental unit of DNA. Now, when it comes down to it, and I have a big picture over here showing how these nucleotides are arranged in DNA, you will see that DNA is like made up of 2 strands of nucleotides. But even more so, one strand runs in a certain direction, which I'll get to in a minute. The other strand is completely opposite of that strand. As you can see from the pictures, the sugars are all pointing in one direction here, the sugars are pointing the other direction here. It's like you've taken the molecules and put them upside down. This is referred to, and this is also in your notes, it should be, as anti-parallel. In other words, the strands run in anti-parallel directions, which is just a fancy way of saying opposite direction. Now, for the process we're going to learn about today, DNA replication, and for that matter, the process we're going to learn about on Monday, which is protein synthesis, it's important that you understand this direction aspect of DNA. So they use these symbols to represent that direction. They say that one strand runs in a 5' to 3' direction. The other strand runs the opposite, the 5' to 3' direction, but it's going the opposite direction. On the board here, I just drew where they get those symbols from, because that was a question in the other classes. Where are they getting these 5' and 3'? You know, if this is more for just your knowledge, they actually number the carbon atoms on a sugar molecule in a clockwise direction. So this carbon would be carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. They refer to this as the 5' end because when a new nucleotide sets its butt down on top of here, it's going to be attaching closest to that 5 carbon, that number 5 carbon. Down here at the bottom, if you see, when a new nucleotide attaches onto this end, it's going to be attaching closest to that number 3 carbon. That's where they get the 5' and 3' terms. I'm not terribly concerned that you understand that detail, but I think it's actually important, or kind of just nice to know where they get these 5' and 3' symbols. That's right. And you can kind of think of it like, well, this one's on the upper left. It's kind of like that's the portion where something's going to attach to. That's the 5' end because it refers to the carbon, the numbered carbon.
So the one with the phosphate, like, okay,
yeah,
I got it.
Yeah, and you can even see on the other strand here, this is the 3' end because look what end's sticking up there where a new nucleotide is going to attach. It's the 3. It's closest to the 3 carbon. Okay, so that's where they get the numbering system from. But if you don't understand that, when you guys get to organic chemistry in college, it's a lot of numbering and structure and knowing how to do this kind of stuff. You know, organic chemistry is definitely no fun. But that's where they get it from. Now, what I want you guys to know is just these strands are running opposite. The reason that I want you guys to know this has to do with a certain enzyme we're going to talk about, who's real picky. That enzyme is picky because that enzyme is picky because it will only work in a 5' to 3' direction. It won't work the other way. So that's why it's important to understand this to begin with. Do you guys remember the name of the enzyme that's like the main enzyme involved in DNA replication. This would have been, the main one. I know you guys learned about a bunch of them. Which one?
DNA primate?
No, that's not the main one, but polymerase. Yeah, DNA polymerase, which you're going to learn is like a head honcho enzyme involved in DNA replication, only works in the 5' to 3' direction. It'll only, in other words, another way of saying that is it's only going to tack nucleotides onto this end. It will not tack nucleotides onto that end. So that basically, that's the reason you need to kind of understand this kind of, these symbols and what they mean. So, okay, let me kind of plug along. Any questions on that so far? All right, let's get to the heart of it. Oh, this is the fun part. If you have different colors, like a couple of different colors, or a pen and a pencil, it might be actually a pretty good thing to have. Just, I think you only need about 2, maybe 3 different colors.
I'll go squiggly line, straight line.
Yeah.
Really strong straight line.
It helps for this if you have just a couple colors for
Dash
today.
line, there we go. I got this. Squiggly line, dash line, straight line.
Yeah.
Is that your symbol? Is that your method?
I feel like I'm good working on the camera. I am.
Method of madness. Hey, Matt's on the TV.
That's a good.
Oh, that's right. You can note in your notebooks that I've, Okay.
You see that, Pierce? She's tampered with our video.
I did.
Our dad is going to be selling everything.
No, actually, the plants are looking good, though, but we're not going to get to those till next week.
Sarah, did you
Some
water?
of the slows are doing good.
Is water good?
I will probably water them over the weekend anyway, so.
With it locking together, they both are shooting the same amount of light, so they both off.
Okay! Before I get into the details of DNA replication. Okay, a little background. You already took this down many chapters ago. Remember when I had you? You don't have to, but if you want to, but it's a good review for your midterm coming up.
When is that?
What does this thing represent?
The last day before break.
The cell cycle. Remember, this little model represents basically all the different phases a cell will go through in its life kind of deal. It doesn't die at the end, it just divides at the end. But remember, the crust only is most of it. What is the time? What does the crust represent? Interphase. And remember, that's not mitosis. That's just the portion of that's basically the phase that the cell spends most of its time in. Now remember, the G1S and G2 are part of the interphase. G1 phase, cells just got done dividing, it's starting to grow again. Something real important happens at the S phase. What is it?
Oh, God.
DNA.
Oh, that sounds familiar. Isn't that what
I thought-
Mrs.
that's not
Hatcher
what
is
we're
about
talking
ready
about.
to talk about? Yeah.
Wait, is that what we're talking about?
Yes.
The S-phase thing?
Absolutely. In fact, way back when we talked about the cell cycle, we just said, well, your DNA is going to replicate in the S phase. And what that means, guys, that has nothing to do with the cell dividing. The only thing you're doing there is taking a piece of DNA, remember they're linear in eukaryotes, and at the end of this S phase, at the end of DNA replication, you're making a total copy of it, so it kind of starts to look like an X. It goes from being linear to looking like an X. And this strand is identical to this strand. That is what DNA replication does. That's going to pave the way for the cell dividing. Now remember, the cell's not going to divide till way over here in both the M phase and then finally the C phase. M stands for mitosis. Please remember that's not when the cell's actually going to split in two. That's just when that's just the processes where it's going to take that now replicated material and separate it to separate parts of the cell. What's the name of the process where it actually is going to split? Cytokinesis. Very good. Cytokinesis is where the actual cell will split into. So how are we doing on this stuff? Are we remembering it?
Good.
Good.
So far, so good.
So far, so good. Okay, so this is what it is. DNA replication, that's the details I'm going to talk about today. The details of that process. I mean, you know what's the end points. All you're doing is taking a piece of DNA and making a copy of it. But there's details. And there's enzymes involved.
Why does everything have to be so?
Because it's science and it's filled with details. Okay, so let's look at the details. I am just going to draw a portion of a DNA molecule. Obviously, your DNA is much bigger than this. and I'm going to label it a 5' end to 3' end on one end, 3' end to 5' end on the other. You now know what that means. I'm going to draw in places for where the bases will be. Remember, the sugars and the phosphates make up the sides, the bases make up the steps, and there's 2 per step. Okay, and I'm going to randomly put in some bases over here. I'm literally just off the top of my head putting down bases. Okay, and you guys know that since I'm referring to a DNA molecule, you already know what's going to be filled in on the other side because one always hooks up with one and one always hooks up with the other. You guys have to remember that your A's and T's will always hook up together and your C's and G's will hook up together. Please do not confuse this with U's. We are only talking about DNA here. Monday, when we talk about the completely other process where we bring RNA into the picture, then you'll talk about the U's. For now, with DNA replication, these rules of base pairs will always apply and nothing else. Okay, so I got my DNA molecule here. Okay, a real little portion of it, very, very small portion of it. By the way, guys, keep in mind, in human DNA, it's going to be 3 billion of these steps long. That's how big our DNA is, 3 billion steps long. Pretty big, pretty big. But we have a lot, there's a lot to us. Actually, you're right. And that's a whole other kind of issue. You're right.
3 billion DNA things in 1 minute.
3 billion. Okay, here's what I want to do. I'm going to talk, now I've got my DNA. Where'd it go? Just got rid of it. I got my DNA. And now let the replication process begin. Very first step, who remembers what happens? What's this thing going to do?
Unzip.
Very good. All I'm going to do on this next slide is take that same DNA molecule and rip it open. And keep it the same on both sides.
Let me spread it out. Just gonna spread it out.
Just redraw. Yeah, just draw it spread out. 1, 2, 3, 4, 5, 6. I think I have 6. Yeah. And just fill it in again as you would. There's a point to this. Believe it or not, it seems like busy work, but it's not.
Same exact thing.
Same exact thing. All you're doing is taking that same molecule and ripping it apart, which is going to bring me to the very first enzyme involved in that process. Just for clarity, keep the 5' and 3' going on. All I did, guys, is take that one molecule and literally unzip it. I ripped it apart. The name of the enzyme that is responsible for doing that is something called, very good, helicase. Enzyme that unzips DNA. I'm going to keep it easy. That is, but by the way, helicase, what kind of bonds are those things breaking?
Hydrogen.
Very good. It's basically helicase is going to be there breaking those hydrogen bonds together, breaking those hydrogen bonds apart, letting the molecule just unzip. Huh?
Those hydrogen bonds are weak.
That's right, because and that's right. Those hydrogen bonds are weak, so this helicase molecule is probably just okay. By the way, another word. I'm going to refer to both of these strands here as template strands. Template strands is kind of a generic word just used to represent like the original strand, like the parent strand. Yeah. These are both, even though they're unzipped, they're both part of the parent strand. They both came from the parent. Okay.
So it's a model for the new one, right?
Yeah, exactly.
Like
So
a
each of
template.
these strands is exactly going to become a model for the next one. Okay.
Question.
Go ahead.
So when they replicate, like the new DNA that's on the left side, is that gonna be the same as the?
It's going to be the exact same thing anyway.
Yes,
isn't
you
it?
want it to be the exact same. So even though they're kind of reversed, they're the same. They have the same genetic information, which is what you want. Exactly, because if I want my skin cell to make another skin cell, the genetic material better be the same in both of those new cells.
Or you're going to get
Yes.
a freckle.
So absolutely. Okay, a little more detail here. It's not that easy. There's going to be DNA polymerase is going to come in here and tack on new nucleotides. However, DNA polymerase is kind of funny. It needs like a primer. For lack of a better word, I'm going to use primer. You guys know when you paint a room, a lot of times you'll put like a primer coat on the wall to kind of get the wall ready to be painted. I don't know if any of you guys paint. Anyway, so you guys paint? It's like primer. You get it ready. In other words, in order for DNA polymerase to be able to come in here and do its work, you gotta lay down
It's
these
like when you start the lawnmower.
primers.
It's like when you start the lawnmower, you gotta press the.
Yeah, that's right. It's a primer button. That's exactly right. On the primer button on the lawnmower. I'm going to represent primers by these little red red boxes. This is going to be a primer here, and this is going to be a primer over here. Now you notice something.
Jesse, do like squares, triangles, and circles.
When I was
Use
using
a
my
squiggly
condo
box.
last week, I laid down 2 coats of primer. Just
Okay.
to make sure.
By the way, there's another enzyme here I want to talk about. There's an enzyme called primase.
Oh, really? So interesting.
Enzyme that makes the primer. And by the way, just so you know what it physically is, now don't get confused when I write this, but just so you know what it physically is, a primer is actually like a short segment of RNA. But please do not confuse this process with Monday. That's the only time I will mention the word RNA, and we're not even going to be concerned about what its like code is.
So should I not even write that?
No, you can write it just so you know what the primer physically is. It's actually a little short segment of RNA. However, the primers are going to eventually be cut out and removed in the end, so they're not going to be part of the final molecule anyway. They're just like a primer. They're like the button on the lawnmower. They're kind of there in the beginning.
Is there more than one of those on each side?
We'll get to that.
Along the whole.
We will get to that.
Oh boy.
One strand will have one primer.
Calm down.
No, but to answer your question, it might make sense from something you've already learned. One strand is going to have just need only one primer, their leading strand. The other strand is going to need a lot of these suckers all along the strand.
I was talking about like along the whole DNA piece, like they started more than one spot, right?
Yes, you're right. So I guess you're right. In one sense, there'll be one at every replication bubble. You're right. On one strand there'll be one, and another one there'll be many. And then there's another bubble that has one on one strand and one on the other. Yes. Ooh, you see, you guys picked up on quite a lot. Believe me. Okay, here's the deal. Now that these primers are laid down, this main head honcho enzyme called DNA polymerase. Now, notice the trend here, by the way, guys. ACE, ACE, ACE. You get the idea. DNA polymerase is going to be the main enzyme involved in this whole process. The enzyme that adds new nucleotides to growing DNA chain. I'll say for lack of a better word. Okay? So it's the enzyme that's going to be adding on nucleotides one at a time. You'll see a video here in a minute. It's just going to boop, 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 one at a time to the growing DNA chain. Here's the catch. This enzyme is picky. It works only in the 5' to 3' direction. Another way of saying that is that it will only add nucleotides to the 3' end only. Only adds nucleotides to the 3' end. I'll show you this all in a minute. This is why I put my primers on separate. One is on the top and one's on the bottom. I've got the 3' end down here. And as you guys know, the DNA that I construct better have the 5' end over here, and it better have the 3' end over here, the new strand. DNA polymerase. By the way, this primer is like a starting point. It is only going to add nucleotides in a 5' to 3' direction. It'll only work this way on this molecule. So that first nucleotide at the bottom, what's it going to be? T. You guys know these, so this should be easy. The next nucleotide it adds is going to be what? C. The third nucleotide, you get the idea. You get it. And the reason I'm drawing it like this, just so you know, it's going to go one at a time. 5' to 3' direction. It's now made a new side of the DNA in the 5' to 3' direction. Bada bing. What'd you say? Do you have a question?
Is there any way that, like, is there a disease where it goes backwards? Is that even possible?
I don't even think it's possible because the way DNA polymerase works, it wouldn't even, and I'm sure the details come down to a chemical reason, but it's only able to link nucleotides on and connect them that way, in that direction. Details, I don't know. Yeah.
Wouldn't that be on the
Huh?
Wouldn't that be going from top to bottom now?
No, you're looking at the template strand. You got to think of it from the other strand's point
So
of view.
whatever you're doing is the opposite of it.
Yeah. It's you got to look at it since
I
you're
thought-
building a new strand. The new strand can only be built in the 5' to 3' direction. Maybe that's another way of looking
that
at
makes
it.
more sense.
Yeah, don't look at the template strand for the direction our DNA polymerase is going to work. We'll look at how basically the new strand and the orientation of the new strand in the 5' to 3' direction. Good question too. Hey, so over here it's going to work from top to bottom, 5' to 3', because your 5' is going to be over here, 3'. So you know DNA polymerase is going to have to work in that direction. You'll see A. You guys know how to do this. T, C, C, G, A. So when it's all said and done, guys, you have two molecules here that are totally identical to each other. Yeah, they're kind of mirror images, but that's okay. They're the same genetic information in the same order. Another word that you guys heard of that I want you to know. This type of replication, big picture here, this type of replication is called semi-conservative replication. The reason for that, semi-conservative replication, semi, half. When you have one old and one new strand in the end, that's considered semi-conservative. The old strand being the original template strand, the new strand being the one that the DNA polymerase set down. So let me kind of draw this like this. It's another way of doing this by taking out some of the letters. When DNA, let's say a DNA molecule looks like this, semi-conservative means that when it's all said and done, you're going to have one old and one new strand. That's different than taking, like if I would take a DNA molecule and literally go put it in a copy machine and get an entirely different strand, that would not be semi-conservative. That would be called conservative replication. But we know DNA doesn't replicate like that. There's been experiments. We know that when DNA replicates itself, it unzips. One template strand for each is basically used as like Jake said, like, you know, the model to build the new one. Yeah.
Since like it saves your DNA, like the old DNA or whatever.
Yeah.
Is that like the reason we go because it gets worn out over time?
No. What happens is this whole process of placing new nucleotides one at a time, sometimes DNA polymerase makes boo-boos. It has nothing to do with how old the strand is necessarily. It just refers to the fact that as we age, a lot of these boo-boos can't get covered up anymore. Too many accumulate, they start showing signs. So, good question too. Wow, I'm fire. All right, so we talked about how many enzymes here? We talked about helicase. We talked about primase. We talked about the head honcho DNA polymerase. All of these are working together to build up a new DNA molecule. There is one more enzyme I want to talk about, and this will lead into another discussion about this leading lagging strand thing, if you guys are confused on that. Okay, I only have one more. I don't have much more notes, though. Okay. Ligase is one more enzyme that comes around in the end. Enzyme that joins DNA fragments. I'm going to actually, you refer to ligase. Actually, the word ligate, I mean, if you know what the word ligate means to begin with, it kind of means join together. Ligase is like an enzyme that's going to act, that doesn't look right, that acts like glue. We're going to be producing, as you guys said, and you're going to have this process taking place in many different areas on a DNA molecule in a eukaryotic cell. You're going to have to kind of find a way for some enzyme to come in the end and kind of glue these pieces together. Now, one strand's going to have a lot more need for ligase than the other strand will, but both strands will need it. Okay, which leads me to this leading lagging strand thing. Okay, like I said, DNA polymerase is funny. It's the enzyme responsible for tacking on new nucleotides, but it'll only tack them on the 3' end. Well, that kind of can create a problem. Let me kind of draw a DNA molecule. I drew a DNA molecule that is in the unzipping process. And I basically, and I'm not going to write in nucleotides for the sake of clarity. All I'm showing here in this picture is the fact that I have a DNA molecule. It is in the process of being unzipped by this wonderful enzyme called helicase, which is at the one end there. You got to kind of picture this thing in motion. This thing is kind of moving. You got your ends of your strands. Let's say this is my 3' end, this is my 5' end, this is my 5' end, this is my 3' end. All I did was do what we've already done. Give directions to the 2 strands. As this thing's opening up the DNA, remember, the top strand up here is going to have an easy time. DNA polymerase, as soon as a new nucleotide becomes exposed, it can tack on a nucleotide. Because remember, the new strand that is going to be made over here is going to be going, and it's going to be being built in the 5' to 3' direction. So DNA polymerase is going to have no problem working this way. That is called the leading strand. And you gotta have to visualize this. The leading strand, because the DNA polymerase, like I said, as new nucleotides are being exposed, DNA polymerase can keep doing its job, tacking them out. However, this other strand's a problem. This other strand, and I know I'm going to get cut off here real soon, guys, because pizza's on the way. Okay. This other new strand, remember, DNA polymerase has to work in the 5' to 3' direction. This other strand is going to be built in the 5' to 3' direction this way, but you have to tack on the nucleotides a little segment at a time. And this is hard to see until you see an animation that I'm about to show you.
Was that what you were talking about, like the
Right, they're broken up somewhere.
This is called the lagging strand because literally they get this word just because it takes longer for this strand. It lags behind. It takes longer for this strand to be built because it has to be built in fragments. Each of those fragments that are being made are called Akasaki fragments. That they just name the fragments. Those are eventually going to need to get glued together by ligase.
Akasaki.
Akasaki, that's the name of the guy who
Obviously
invented
a Japanese scientist.
Yeah, it's a Japanese scientist who basically figured out that this is how DNA
Are they all the
replicates.
same length?
That's a good question. I think so. I think they would be all the same length.
That's so stupid.
But it has to be built this way because basically this bottom strand has to unwind to a certain degree before DNA polymerase can come in and make a little fragment. Then it has to wait for a certain more time to go by for this to unzip more, then come in and build the fragment. Then it has to wait for it to unwind. Did you get the idea?
Yeah.
For it to build another fragment.
Oh, then she's the other side. Why are they?
So it's like the slow kid when they're running.
I understand.
Yeah, it's almost when you see it in an animation, it'll make more sense.
Actually, I'm normally
One
good
of
at
these
those
is like, that's start to go this way.
Correct.
Has to wait for a Friday.
I'm up with you 90%.
Correct. Exactly. And so primers are going to be laid down each time. So you'll have a primer here, you'll have a primer here, you'll have a primer here. Whereas on this strand, you start off with one primer and you just go.
That makes a lot of sense.
Does that make sense more than? Okay. Good.
Oh.
I don't know if I'm going to have time. Has there been people poking their head in here?
I saw Heidi on my lawn.
Okay. I think, yeah, this will be good. Let me see if I can squeeze in one video. She said 10 minutes, and I think we have about 12 minutes. Just to show you this process in action. If you wanted, and seeing it in
How do we
action,
get pizza? I want to know.
they're making it in their food science class, and you guys are their taste testers.
Sweet.
Oh, yeah.
Huh?
Yeah, so who's the best teacher ever?
I thought you like.
Miss Heidi, yeah.
I thought you ordered us pizza. I was like, yeah.
No, I did not order you pizza. I don't love you that much.
Why are they making pizza?
All right, hold on, food science. All right, real quickly, let me show you this. This is not long. This is kind of nice because it's
Check
going to
it out.
show you.
Isaiah, you're gonna watch the movie.
I would give you
So everybody say hi to Isaiah.
Cute.
oxide fragments.
Okay,
That might be a cool one.
this is kind of neat. This is kind of showing in motion both your leading strand. Now this is opposite me. This leading strand is going to be put on real quick. This red blob is DNA polymerase, the main head honcho enzyme. The green thing is helicase, unzipping it all. You do have a couple of other, you have primase here, which is basically going to be laying down primers, continuing on the lagging strand. But it's not the greatest picture, but it's real quick too. I wish it would just keep going and going. But they show the RNA polymerase bringing in nucleotides, but the top strand is done in fragments. Yeah, they don't show it long enough. Let me show one other video that just concentrates on the lagging strand only. Yeah. I like this video. I guess it's a little better because they spend a little more time, but they show that.
Eventually.
Hold on, I will. The lady's talking, but the lady's voice is insanely crazy. It drives you nuts. So I muted her out.
Can we hear it?
Do you want to really?
Yeah, I do actually.
I'll rewind it in here, and this will be the last thing we hear.
Strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together.
We're just going to show the lagging strand.
We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red,
This is important
forms the
stuff.
beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction.
So it just picks random spots?
That's the detail I don't know. I'm not
The
sure
resulting
how it chooses
segments are
parts.
called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand.
By that time, the leading strand's going.
Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding protein, shown here in blue,
Hey, shut her
and
up.
makes an RNA primer to begin an Okazaki fragment.
No, I want to keep listening.
But this is neat. It is showing, now it's showing the details of DNA polymerase coming
The
in and tapping
assembly
on the
process
nucleotides.
continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments.
So basically, the polymerase has to wait for a primer to get
Correct.
before it.
So it's just in there
Absolutely.
waiting.
Exactly. It needs something to build upon.
Instead of working on it continuously like this.
After the
it
fragments are made,
this,
another kind of
this.
DNA polymerase replaces
Correct.
each RNA
Correct.
primer with
You
DNA.
got it. Does everybody get this kind
Next,
of
the
sort of?
enzyme
Okay.
DNA
Is
ligase
this making a lot more
links the
sense?
Yes, I
Okazaki
understand this now. I'm not
fragments to form the lagging strand.
Okay. How we doing, Bryce? You lucked out.
You doing good?
You missed a good day.
Yes, just like me.
We
Yeah,
missed
good
good
thing
days.
she's in good
I picked
mood
good
to
days.
review this stuff. Goodness,